Hello, my name is Peng Wang from Nanjing University. I will be presenting low-dose electron ptychography for cryo-biological imaging. This is a diagram of optical configuration for cryo-ptychography. We operate in scanning diffraction mode. We use pixelated detector to acquire a diffraction dataset. The sample was vitrified using standard procedure and then transferred to the microscope, kept it at liquid nitrogen temperature using cryo holder. Then we use ptychography iterative algorithm called ePy to reconstruct objects. The sample we used in this cryo experiment was rotavirus. This is a conventional defocused cryo-TEM image acquired at dose of 35 electron angstrom square. This is ptychography reconstructed face of the object at a dose of 23 electron angstrom square. In this enlarged image, the face shows stronger contrast from viral particles, and both capsid trimers of viral protein VP6 and the channel between those can be clearly seen. Most importantly, we are able to see the contrast from the overall the shape of the particle, which contain low frequency. To confirm it, we calculate the Fourier transform of two images. As you can see, ptychography has a stronger transfer at low frequency, which can be extended to the size of the viral particles. To understand why the ptychography phase contrast is stronger and better, we may first step back and see the contrast mechanism of defocused phase contrast imaging. This is the stained adenovirus to show you the size and the shape of the particle. However, in bioimaging, people use unstained condition to preserve the native state of the sample. However, due to the weak scattering of the biomaterial, the contrast is really low at low dose conditions. In order to increase the contrast of the particle, we normally increase the defocus value to push the CTF back to the lower frequency, for example 1/10 nanometer, where we can see the shell thickness of the viral particle slightly better. But this causes the quickly oscillation of CTF at high frequency and scramble high resolution information. Therefore, there is a trade-off between the resolution and the contrast in the TEM. Therefore, we need a more ideal imaging system that can not only cover the lower frequency of overall the shape of particle, but also preserve high frequency much better without such oscillation, which can be simply intuitively interpreted. This slide just show you again we can simply increase focus to a very large value to enhance the particle shape. However, the first crossing point has moved to 9.5 nanometer, beyond which you have dramatically oscillation of the contrast function. Therefore, it becomes really difficult to interpret high spatial frequency. This is ptychography reconstructing the phase of the same sample using 1.37 milliradian converging angle. It shows overall the shape of the particle with really good contrast. It has a cold shell structure with hexagonal shell matching the capsid coating and a triangular core corresponding internal genome. The power spectrum of this image is continuous over its entire spatial frequency range and does not show the Schönring-like pattern. In order to understand why ptychography has a better phase contrast than TEM contrast, we numerically calculate phase contrast transfer functions with the Poisson noise as a function of different converging angles. The calculation method can be found in SI in this paper. In comparison to TEM, we have three takeoff methods. Firstly, using lower converging semi-angle, for example 1 milliradian here, the CTF can cover a wide range of the spatial frequencies, and that is lower enough to reveal the overall shape of particle. Secondly, unlike a TEM, The ptychography phase contrast function does not have a zero crossing in entire transfer range, and that is consistent with the previous observation, which means ptychography phase doesn't have contrast reversal over the entire spatial frequency. Therefore, the resulting contrast can be directly and intuitively interpreted. Finally, we can change convergence semi-angle to shift the bandwidth of the contrast function from lower frequency to high frequency. And this is a significant advantage for ptychography because CTF can be tuned to enhance the information transfer for a particular spatial frequency range for the target object. To further demonstrate the tunable bandwidth of ptychography contrast transfer function, we reconstruct a phase of an adenovirus particle at different converging angles from 1, 5, and 10 milliradian. We use Fourier ring correlation with half-bit criteria to evaluate resolution. The results show that the resolution are increased as the converging angle increase, which consistent to the theoretical prediction. Recall that when we use converging angle up to 24 milliradian, we were able to reconstruct atomic resolution of molybdenum in material science. Therefore, ptychography information transfer can be tuned to different information frequency range. Furthermore, as we use relatively large defocused probe in the experiment, it is possible to scan a large sample area with a small number of probe positions, which further reduce the total dose. Together with ptychography's high contrast, it can still clearly show the viral capsid shape at the dose of 5.7 electron square. Another example shown here is HIV particle at very low dose. We still can observe the particle at 5.7 electron square. Ptychography reconstruction is very dose efficient and can be applied to biological molecular imaging in cryo-states at very low dose. As scanning mode in nature, ptychography reconstructed area can be theoretically unlimited. Here we demonstrate 1 micrometer squared area from adenovirus infected cell reconstructed at dose of 27 electron square. The phase show high contrast of ultrastructural features with the different sizes. Therefore, we believe ptychography can be used to image very large biology objects, potentially at cellular level. I think it is really an exciting time for electron ptychography as it has shown many advantages. For example, light atom detection, low dose integration with yields, optical sectioning, and tube tomography, which are demonstrated in material science. Now, cryo-bioimaging for structural biology as presented here. Therefore, we believe there are many opportunities for electron ptychography to implement in both material science and biology research areas. In summary, we have demonstrated first cryo-electron ptychography with high contrast and at low dose with very large field of view. And this approach is suitable for biology molecular imaging. We also shown this approach have tunable continuous wideband information transfer, especially include low spatial frequency, which are inaccessible to conventional phase contrast imaging. We anticipate by combining ptychography with single particle analysis, we should be able to reconstruct three-dimensional small molecules with low symmetry, low molecular weight, or reconstruct larger scale molecular architecture in individual cells and multicellular organisms. And this work is a result of the close collaboration among people on this page. Okay. I would especially like to thank Angus Kirkland and his group from EPSIC and the Rosalind Franklin Institute for their great support and contribution to this project. Without them, it wouldn't be possible to accomplish this work. I would like to thank Jingdong Song from Chinese CDC, Peijun Zhang's group from EBIC, David Stewart Group from Oxford Structural Biology for providing biology samples. Finally, I would like to thank Peter Nellis from Oxford University and Xiaoqing Pan from UC Irvine for their strong contribution to this work. Thank you very much for listening.